In this video, I'm going to help you understand how the electrical activity and the chemical messages are sent from one neuron to the next. I'll come back to the gray matter and white matter idea in a little bit, but for right now, let's look at this. Cells produce electrical signals called action potentials. An action potential really just describes this wave of electricity that sweeps down the neuron. The electrical activity is passed from one part of the cell to the next because of the movement of ions in and out of the cell. So think about this. Ions have an electrical charge, like this chloride or this sodium. If I concentrate the ions on one side of the plasma membrane and then allow them to sweep in and out, it will cause a change in the electricity that we could measure if we had a tiny detector. So what we're going to see is a graph that looks something like an EKG, something like a heartbeat. That is representing the change in the voltage, the change in the electricity as time goes. So if I park my little detector right here, we're going to see a wave sweep down, and that wave will measure this up and down of depolarization and repolarization. You can watch these videos. I'm sure you'll find them to be really helpful. And we have excellent videos in the Blackboard Chapter 11 folder that you can watch as well. So here's how it works. At the baseline, the cell is polarized at a negative 70 millivolts. This is a tiny amount of electricity, right? A 9-volt battery on my tongue just barely gives me a little charge. We're talking about negative 70 thousandths of a volt, so it's a really tiny, tiny charge, but it is less than zero. It's negative. Why is it negative? Well, because of this idea of resting membrane potential. At resting membrane potential, negative 70, a little pump has pushed all of the sodium out of the neuron and pulled all of the potassium in. In order for the sodium to leak in, it's going to have to use a sodium channel. And in order for the potassium to get out, it's going to have to use a potassium channel. But because we know the plasma membrane is impermeable to these ions, and at this point all the doors are closed, the sodium can't get in and the potassium cannot get out. It's also true that there are very large negatively charged proteins on the inside of the cell. You might think about transcription factors and various signal molecules. It is really the presence of those large anionic proteins that make this such a negative number. But if you read the book, you'll also see there's various variable permeability to potassium. Anyway, negative 70. In the event that we can open some of these doors, we'll allow a little bit of sodium to leak in. And if we get enough of it leaking in, we'll reach this dotted line called threshold. Threshold's at negative 55, and that's a very special number. The reason is most of these doors, most of these voltage-gated sodium channels are designed to open at negative 55. It's a really key concept to this chapter, and it has to do with the trigger zone. If we go back here, remember, if we stimulate this thing a little bit, nothing happens. But if we stimulate it enough, it's going to fire. That number, negative 55, is what's going to open these doors at the initial segment and cause the signal to travel full strength, full speed, all the way down to the end. Here's what happens when we reach threshold, this idea of the all or nothing principle. If we don't reach threshold, nothing. If we do reach threshold, all. Full strength, full signal. and it's because of the opening of these voltage-gated sodium channels. Once they open, it allows all of that sodium to rush in. We know sodium has a positive charge, and so we see an upward deflection, an upward movement. That's called depolarization, right? And that word means we're getting less negative. We're getting closer to zero. You see, we can actually overshoot a little bit up to positive 35. Positive 35 is the perfect electrical signal to open the voltage-gated potassium channels. When they open, the potassium rushes out, taking with it that positive charge. And look at this, really steep, almost straight down. Because we're becoming more negative during that outflow of potassium, we call this repolarization. Because we've gone back into negative territory, we are once again polarized. As a matter of fact, we go beyond negative 70. And so this phase here is called hyperpolarization. We're beyond resting membrane potential. And in order to get back from this little trough here, back up to resting membrane potential, we have to close the potassium doors, and this sodium-potassium pump is going to push all of the sodium back out and pull all of the potassium back in. And once all the doors are closed, we go back to resting membrane potential. Okay, so this is really complicated, but super important. It helps us to understand how the electrical signal is sent, and it'll help us to really see as we move forward when the electrical signal is sent and how neurogenic disorders or spinal cord injury can interfere with the signal being sent. What I recommend that you do here is get out your Play-Doh or your beads, get out your markers and your paper, and reproduce this. Just looking at it and thinking about it isn't really going to make it stick in the same way as if you draw these out and to make them happen. So that'd be my advice. Make 5 or 6 flashcards. Make sure you can put these in order. Make sure you can describe which ion is moving and in which direction. For example, you could imagine a question like this. During repolarization, which ion is moving and in which direction? During repolarization, potassium is moving out of the cell, which is why the electrical activity goes back into negative territory. Okay, the refractory period is really this idea that the neuron can't be stimulated to have another event until it's finished having the first event. You might recognize this phrase from human sexuality, right? After a climax, there is a period of time during which a person can't have another climax. And although the period may be variable, we use that same term. Think of it like a sneeze. If we sprinkle a little pepper on your nose and we reach a certain point, you're going to have a sneeze and you can't stop it. You're going to have a full sneeze and then you're going to come back to normal. Can I sprinkle more pepper to have you have another sneeze somewhere in this purple or pink area? Nope, because you're already having a sneeze. You got to get back to normal before you can have a second sneeze. There's another example. A neuron cannot have another action potential until it's done having its first action potential. If you want more detail, the absolute refractory period really refers to the amount of time it takes to close the sodium channels. And so no matter how hard you stimulate this neuron, the opening and closing of the voltage-gated sodium channels is occurring during this period. And so you could not stimulate this neuron to reach threshold again. The relative refractory period really lines up more so with the opening and closing of the potassium channels. And so theoretically, with enough stimulation, you might be able to get this back up to threshold to have a second event before the end of this pink region. However, because the sodium and potassium are on the wrong sides of the plasma membrane, and because it results in a hyperpolarized state, we're even that much further away from threshold. So it's very unlikely that a neuron could be stimulated to have a second action potential until we finish the relative refractory period, restoring resting membrane potential. How about that? Okay, you should look at all of these terms, but the most important ones are really the first, threshold stimulus and subthreshold stimulus. Here's what it means. Subthreshold stimulus means maybe you have a little pepper on your nose or there's a little tickle on your nose, but it doesn't cause you to sneeze. If you do get up to threshold, it's going to cause the event to happen. At the cellular level, it's very similar, although it's not going to give us necessarily a conscious awareness of that tickle. If we stimulate one neuron just a little bit, the sodium-potassium pump will restore resting membrane potential and nothing happens. If we do reach threshold, it will send a signal. And so we might think of this as just a little click in the nervous system. So if there's a tiny bug on your hand and you just barely feel it, if the bug moves a little bit, you might get a stronger stimulus and you get a couple clicks. When the signals are firing really frequently and constantly, that's what gives us a very strong sensation. So let me say all of that in one short sentence if I can. The increasing frequency of action potentials is what tells the brain how strong the stimulation is. A little itch sends a signal every so often to the brain, and I can feel it, but it's not a strong stimulus. If someone jabs something into the back of my hand, however, man, that neuron's going to fire at a very high rate. And so the increasing frequency of incoming signals is what tells my brain how strong the flavor, or how loud the sound is, or how painful. Propagation of action potentials means how does the signal travel down that long fibrous axon? And it really comes back to this flux of ions. If we reach threshold, we get sodium to flood in, we get potassium to flood out. That change in ion movements causes an electrical flipping, that polarization, right? Poles plus or minus. And as that wave moves, it triggers the next voltage-gated channels to open, and the ions flow in and out, giving us that flip in polarization. So if you watch those animations, the links I have for you, you'll really appreciate how this moves down the axon. Now, in reality, it travels at a very fast rate. In an unmyelinated axon, maybe a few meters per second. In a myelinated axon, it travels way faster than that. And so at 6'1", like myself, if the signal's traveling 30 or 40 meters per second, it goes from my toe to my brain in a fraction of a second, actually less time than I can really perceive. And so I perceive it as being an instantaneous reaction. Before we move on, I want to point out one other key. This wave always and only travels in this direction. On a multipolar neuron, anyway, it comes from the dendrite to the cell body, down the axon, and then to the end. Once you start the signal at the trigger zone, it will always fire in this direction, and the signal can't bounce back in the opposite direction. And this is why. The absolute refractory period prevents another action potential. And so as it moves from left to right, the wave of depolarization, shown in orange, is always followed by a wave of hyperpolarization, the absolute refractory period. So the signal will always and only go in that direction. Myelinated axons are so fast because instead of depending on that wave of depolarization to move segmentally down the axon, it gets to the myelinated section, and then diffusion of the ions actually goes even faster. And so it triggers the action potential, and then that triggers the next action potential. So In other words, it's almost as if it travels from here to here in no time. Here to here in no time. Here to here in no time. And so if we had an unmyelinated axon just below, you could imagine that this one would be to the end before this one had even just barely started. Now we're going to talk about how the neurons communicate. As strange as it sounds, one neuron doesn't actually touch its neighbor. They just come ridiculously close together. Remember the idea of the perfume bottle and this stuff getting sprayed out the end? It is sprayed into a tiny space called the synaptic cleft. A cleft is a tiny break or a tiny space. And the word synapse refers to a connection. A chemical synapse is a connection between two cells where a chemical is released by one and detected by the other. And when it's detected, it causes a change in the second cell. In this case, the chemicals that are released are called neurotransmitter. Dopamine is a neurotransmitter, and so is GABA and serotonin and acetylcholine and epinephrine. Many different types of neurons, and each releases its own sort of flavor, its own odor of neurotransmitter. The cell, excuse me, the cell on the other side of the synaptic cleft is called the postsynaptic neuron, and if it wants to pick up the whiff of the chemical, it has to have a receptor. And in this case, the receptors are the end of a larger structure, a doorway, called a ligand-gated sodium channel, or ligand-gated sodium channel, if you prefer that pronunciation. In this case, the ligand is the chemical signal. And we know that a gated ion channel means a doorway for ions that can be opened or closed. In this case, it's closed, keeping all of the sodium on the outside of this cell. But as soon as the chemical fits into the keyhole and opens the door, the sodium rushes in, and we're going to see depolarization, just like we had learned before. Sodium rushing into the cell causes this cell membrane to become positive, and if it reaches threshold, it's going to fire, and the whole process repeats. Although this process might be a little bit complicated, this is what you need to understand. With the arrival of the action potential onto the synaptic knob, or presynaptic terminal, voltage-gated calcium channels are opened, allowing calcium to rush in and cause synaptic vesicles to exocytose their neurotransmitter. The neurotransmitter flies across the synaptic cleft in almost no time, binding tightly to the receptors on the ligand-gated sodium channels. When those channels open, sodium rushes in, causing a local depolarization on the postsynaptic neuron. Thank goodness there are helpful YouTube videos so you can walk through and learn how this works. In case you're wondering why you need to know how this works, let me remind you that many of our meds work at this level. Selective serotonin reuptake inhibitors, for example, antidepressants, anti-seizure medications, medications for heart rate and blood pressure, and medications for pain. Many of these work at this level, either binding to the receptor with a chemical and locking the door shut or locking the door open, or in some cases, cutting up the neurotransmitter or allowing the presynaptic neuron to suck it back up or to prevent it from sucking that neurotransmitter back up, which is what a selective serotonin reuptake inhibitor is all about. Okay, so here's my point. This might be complicated and boring, but you are going into medicine and you need to know how medicines work. And if you want to know how pharmacology works, you have to understand the chemical synapse very, very well. When the neurotransmitter is released into the gap, we want it to act, and then we want it to get the heck out of the way. So there are a couple of different ways that we remove the neurotransmitter, and they work almost instantaneously. The first is by using an enzyme like this one. Acetylcholine, ACH, is a specific neurotransmitter. The enzyme acetylcholinesterase is the enzyme that splits it apart to prevent it from continuing to stimulate the second neuron. Other examples allow the reuptake, the recycling of the chemical back into the presynaptic neuron, or by allowing the neurotransmitter to diffuse away from the synapse so that it can no longer bind. These are some visuals showing how that process could happen. Above, we see the enzyme breaking down the neurotransmitter and then slurping up the components to recycle them to release it next time. And in some cases, it's just sucked right back up anyway. The next concept we need to understand is that some of our neurotransmitters excite the postsynaptic cell, and others inhibit it. Excitatory neurotransmitters increase the chance that the second neuron is going to fire and that the signal is going to be sent on. Inhibitory neurotransmitters tell the postsynaptic neuron, shh, don't fire, don't pass the message along. Now think about it. This is actually a lot like a computer circuit. The ones and zeros of the binary code are really just tiny little electrical excitations and inhibitions, tiny switches. And in very much the same way, many of us have come to think of the brain as a biological computer, using excitatory and inhibitory signals to have a binary code that creates human consciousness. Here's how we would graph the effects of an excitatory neurotransmitter. Like the animation we had seen, or the illustration we had looked at just a few minutes ago, when we open the sodium channels, it allows sodium to rush in, causing a little local depolarization. This is sometimes called an excitatory postsynaptic potential, but it really is another example of a subthreshold stimulus. You can imagine during this short period of time, this cell would be more easily brought to threshold. And so if there are other neurons coming in, or if there are sensory receptors attached to this neuron, it only has a little distance to go before it would fire. Inhibitory neurotransmitters cause an inhibitory postsynaptic potential. And it just means a little short-term hyperpolarization. During this period of time, even if we sort of tickled that dendrite, it would be really hard to let enough sodium in to reach threshold. And so this neuron is probably not going to fire. Let me give you specific examples, big picture examples of why we need these two different processes. If I have a question and I want to raise my hand, I have to excite the neurons that go to my arm and extend my elbow and flex my shoulder to stick my arm up in the air. But I also want to inhibit the other muscles. I want to inhibit my biceps, for example. Otherwise, I'm going to stick my elbow up in the air. And I need to inhibit some of the deltoids. Otherwise, my hand might stick up, but my arm's going to just sort of shake and flex next to my body. Okay, so here's the point. Sometimes we need to excite one muscle and inhibit the other to allow for an easy and fluid movement. What about thoughts? If I'm trying to write an article or trying to read one, I want to be able to focus parts of my brain to think about the words and ideas, but I want to try to turn off my other thoughts. I'm thinking about dinner, or I'm thinking about going outside for a walk. No, I've got to inhibit that thought so that I can get my work done. Or if I'm in a meeting and I have a thought start to rise and I'm going to tell off my boss, but my other part of my brain says, hey, dude, you need this job. You better smush that thought. So, is an excitatory signal more important than an inhibitory one? No, they're equally important. There are times when we need to excite the neuron, and certainly times when we need to inhibit one. My last example, in cases where there's too much excitation, it could lead to a seizure. So we might think of a grand mal seizure as a really dramatic example of too much excitation, right? Where there was sort of this uncontrolled electrical storm going through the brain. Or we might think about other types of seizure or narcolepsy as being too much inhibition. I once had a colleague who used to fall asleep during his patient consultations because he had narcolepsy, and there would be too much inhibition in his brain, and in the middle of a sentence he would just sort of black out for a little bit, leaning his head on his chin, and then the patient would say, "Doctor? Doctor? Oh, yes." So he wasn't really asleep, but anyway, another example. Too much inhibition can cause altered states of consciousness, altered sleep-wake cycle, altered sensation or paralysis. Here's a good way to think about the ideas we're learning about here. We can see that there are other cases where we might try to prevent this one from firing by releasing what we call a presynaptic inhibitor. In other cases, stimulating it with a presynaptic facilitation. An interesting idea, not a really super important one. These ideas just refer to the fact that we can have a lot of neurons connecting in a very small area that could lead to threshold, or we could have a lot of stimulation in a short amount of time causing us to reach threshold. Either way, we have to reach threshold by sort of adding up the stimulation, summation. And that can happen either spatially because of very close proximity to the trigger zone, or with a high frequency of firing causing summation within a short period of time. That's what that word means, temporal. Okay. This one is kind of cool. It's a little complicated, but think about this for a second. At the neuronal level, your brain and consciousness is really like a voting machine. There are yes votes saying, yeah, you should totally fire. There are no votes coming in saying, no, you shouldn't do it. And this other one saying, yes, you should fire. And this other one says, no, you shouldn't. This one says, yes, you should. So all other things being equal, and imagining that all of these signals arrived simultaneously, we have 1, 2, 3 yes votes and only 2 no votes. And so maybe we'll reach threshold and maybe we'll have an event. Let me give you a real life example. What if this is a key neuron deciding whether or not I'm going to eat a donut? This one says, I'm hungry. Yes, you should totally eat that donut. But this one says, yeah, but you're kind of chubby. You better not do it. And this one says, hey, you worked really hard today. You totally deserve it. And this one over here says, yeah, but you might get diabetes. And this one says, hey, it's 2 for 1. Yeah. So then I eat 2 donuts. Another way of thinking of it is sort of like the old angel and the devil in the cartoons, right? What that really represented is that oftentimes we have a couple of different interests in our own mind, right? I am interested in that donut because I am hungry, but I have a poor self-image and I know I'm a little overweight. And so these two ideas are sort of fighting it out in my mind. In very complicated ideas, including religious ones and political viewpoints, man, there are so many different competing interests that can cause us to sway one direction or another. A couple of basic neural pathways that we have to look at here. Convergent pathways start with many, many different impulses, and then through decision making, they yield sort of a single output. This is whether I should eat the donut or not. I'm hungry, I'm chubby, I'm afraid of getting diabetes, I worked really hard, and then they're going to decide. A divergent pathway usually starts with a small stimulus, and this could be an internal or an external one. But through this branching and stimulation of multiple neurons, we end up really having a lot of different responses. So maybe this is the guy out on the trail and he sees the bear, and this is his autonomic response. Okay, stimulate the heart. All right, let's get our blood pressure going. Let's increase our respiration and shut down digestion. Reverberating circuits have this very strange nature of looping. And so depending on the amount of inhibition coming in on these other neurons, this is how things can get sort of stuck in our head. So a particular song or a particular thought that we keep reliving or we would prefer to not experience. It just keeps looping. These are not always annoying or pathological, by the way. Oftentimes, really refined motor activities, a professional piano player or a professional ballerina, they have to have some of these reverberating circuits as well, which make for really, really efficient movements.